Hi, welcome to this video where we're going to do a run through of pretty much all the things you need to know about enzymes, and that's for OCR A-Level Biology Module 2.4. So I've sent you on Teams these two versions of the pictures that we're going to be labeling and using. First of all, right here is the version if you happen to have A3 printing ability, and then of course there's this one, which the image is a little bit larger, which you should first of all download, print, cut out all the pictures, and then you're going to use those to make your A3 mind map. Okay, so this is the way it's going to be laid out. We're going to be talking about enzymes, how they work. We're going to be talking about temperature, pH, how they affect enzymes. We're also going to be talking, looking at lots of different graphs to do with enzyme activity, and how that's affected by inhibitors or by substrate or enzyme concentration. Okay, so if you need to pause the video and get all your stuff ready, do so now, and then we're going to get started. Okay, so first of all, let's get straight into it and talk about enzymes. That's what we're learning about today. Enzymes. Let's make a big title up here. Okay, so what are they? Well, enzymes, first and foremost, they are biological catalysts. So biological catalyst means that they are proteins, and they have a complicated 3D structure, so tertiary or quaternary structure. That's the symbol for tertiary or quaternary structure. And they work by lowering the activation energy. So they lower the activation energy of a reaction, which speeds up the rate of reaction. So that's some just stuff on catalysts in general. They speed up a reaction, but they aren't used up. So let's dive in a little bit more into this diagram here, and let's look at the different parts of it. Okay, so here we have the reactants, and here we have the products. In this specific diagram, we can see that we've got a molecule that is being split into two. So this would be like a hydrolysis reaction with an enzyme catalyzing that reaction. Okay, so here we have two things labeled. So this is the energy level of the reactants, sort of here, and this is the energy level of the products here. Now actually we can see that this is an exothermic reaction because energy is being released. So we're going from reactants down to products, so I'm just going to label that. So that's the energy change. We could say exothermic, just to bring in a little chemistry. But the thing that, that we need to focus on here are these two different heights. Okay, this is the activation energy, which we give the symbol EA, and so is this. Okay, so this top one, EA, is the EA, use black, actually, no, use red. So EA uncatalyzed, so I'll just write uncat, hopefully you can see that, uncat. So this is the activation energy for this reaction in an uncatalyzed manner without the enzyme. This one here, which I'll label in blue because this line is blue, this one is the activation energy which is catalyzed. So there is a much lower activation here, catalyzed, via the catalyzed pathway, which means that more molecules have enough energy to react. So the reaction happens faster. Okay, so this is I'll label here. So quite often we have an alternative reaction pathway, okay, which means that the reaction can be sped up. Okay, so how much do they speed up the reaction? They can speed up a reaction up to 10 to the 12 times. Is that a billion? No, that's more than a billion. That's a trillion 10 to the 9 billion, 10 to the 12. So they can speed up the rate of reaction up to 1 trillion times, all whilst working at normal temperatures and pressures. So basically, most chemical reactions in a living thing are catalyzed by enzymes, pretty much. Almost all of them, we need enzymes to make these reactions happen fast enough to kind of support life. So they are really, really effective at speeding up reactions. Now, in terms of the method, let's go to that next. Okay, so there's two models. Models for enzyme action. Okay, how do they work? Two models. Okay, first one is called the lock and key model, and the second one is called the induced fit model. Now, in reality, they're kind of similar. If we just zoom in a little bit more, we can see that here we've got, in this example of an enzyme here is joining two substrate molecules together. So enzymes can be kind of enzymes that split things apart, or they can join things together. So in this case, joining things together. So we've got two substrates here going into the active site of the enzyme, which is here. Then they temporarily form the enzyme substrate complex, where the substrates are kind of bound to the enzyme. And that's very short-lived state. The enzyme is unchanged, and the product is then released. Okay. Now, the induced fit model is exactly the same, except you can see that there is a subtle change of shape. There's a subtle change of shape when the substrates come and join with the enzyme to form the substrate complex. So let's kind of label that there. So it's a bit like, it's been described as, it's as if when you put a glove onto a hand, it slightly changes shape to fit the hand. Okay, so there's just a slight conformational change between the enzyme and the substrates, kind of just fitting together a bit like that. So there's a subtle change of shape. And in the lock and key model, there is no change of shape. The enzyme is already the perfect shape to fit the substrates. Okay, so that's really the difference. Okay, and in each one, the enzyme is released and is kind of back in its original shape. So that is the same as that, and this is the same as this, and the product is released. Okay. What else? Okay, now let's talk about what other things, or where enzymes can work, and also how they work. Okay, so a little bit more detail. Let's give this the title. When and where? I guess when and where. Okay, so, well, really when is all the time. Enzymes are working all the time throughout your body. Where, I suppose, is really what we want to talk about more. And we can say that enzymes can work intracellular. Well, they can be intracellular. I suppose you would say they can work intracellularly or extracellular. Okay, so they can work inside and outside a cell. For example, intracellular, let's give an example of an enzyme that is intracellular. A good one is example is catalase. And that actually, if you want to go a bit more detail, it takes H2O2 and breaks it down into H2O plus oxygen, like that. Okay, that's supposed to be a bracket there. And hydrogen peroxide is produced in cells all the time, quite often by respiration, and it needs to be broken down quickly because it's toxic and it can damage the cell. So most living cells contain the enzyme catalase to break down this hydrogen peroxide, and that happens inside the cells. Also, we can have extracellular enzymes. For example, let's talk about proteases. There's different types of proteases that work in human digestion. So in human digestion, the enzyme pepsin is produced in the stomach. So pepsin is produced by glandular cells around the stomach, and it's released into the stomach. So when it's operating in the stomach, that's not inside a cell, it's outside a cell. So that would be an extracellular enzyme. So quite often these extracellular enzymes are often digestive enzymes. Digestive enzymes, proteases. Also remember that things like fungi, they produce extracellular enzymes because they grow, they're mycelium threads, a bit like my fingers, they grow out through a substrate, they release the enzymes out of the cells, which digests the material outside of the fungal hyphae, and then it takes the fungi kind of absorb the nutrients back in again. So I'll just put a little note to that. So digestive enzymes, and then we'll just put a little note also, fungi, etc. Okay. All right. So intracellular and extracellular. The other thing that we need to know is some enzymes need an extra kind of piece to be working. So what color shall we use? Let's use red. So some enzymes need something called a cofactor. So cofactors is basically any non-protein substance that has to be present for an enzyme to work. Okay, so let's make a note of that. Non-protein substance that must be present for the enzyme to work. And we divide cofactors, the group of things that we call cofactors, we divide that up into two separate parts, okay? So some cofactors are bound to the protein tightly. So they're basically always there, and we call those prosthetic groups. Other cofactors aren't always bound to the enzyme, and they kind of float in and bind just before it kind of works, and then they can float out again. And we call those coenzymes. So we distinguish between prosthetic groups and coenzymes. Both of those things, prosthetic groups and coenzymes, grouped together we can call cofactors. Okay, so the definitions can be a bit tricky there. So this diagram here shows that. So we've got prosthetic groups and we've got coenzymes, okay? Two different types. So let's try and label prosthetic groups is more permanent, and coenzymes are temporary. Coenzymes often can be ions as well. And there is one that you need to know. Two examples. Let's just write examples. Zinc is an enzyme, sorry, not an enzyme, is an ion as required as a coenzyme for carbonic anhydrase. And chlorine is required for amylase. And those are the two examples that you need to know, okay? Right, moving on. Okay, so just before we move on, actually, I just remembered something I wanted to add. Enzymes, they can speed up different types of reactions, and two types of reactions that you want to know are, so we can have anabolic reactions, which are building things, so joining things together. So these enzymes here, this in this kind of models picture, these are anabolic enzymes because they're joining things together. And we can also have catabolic. So you should just be aware of those two words. So catabolic is breaking or splitting. So in the two diagrams that we've looked at here, this here would be a catabolic reaction because it's taking something and splitting it up, and this would be an anabolic reaction. It's also worth saying that they tend to end in A's, so A's, because things like amylase, lipase, protease. If it says ase on the end of it, it means breaking it down. And we've got different types of enzymes as well. I don't know where we can put this. Maybe we can put this over here. Other common types of enzymes, you can have hydrogenases, tend to add hydrogen. Decarboxylase, this is going to be relevant more in year 13. That would take away a carboxylic acid group or a CO2 molecule. And we've got loads of different, basically normally the enzyme tells you what it does, like peptidyl transferase, for example, which we met last Mindmap, moves a peptide group from one place to another. So that's just one more thing on enzymes. Okay, now we're going to look at things that affect enzymes. So we're going to scroll down a little bit. We're going to come down this way. And we're going to zoom in a little bit more. So first of all, let's look at temperature. Okay, and let's make it quite big. So temperature affects enzymes. Okay, quite a lot. So here is a typical graph showing the effect of temperature on enzymes. And actually, this is not a typical enzyme, because this enzyme has its optimum here at about 55 degrees Celsius. That's quite hot. So let's just say that normally the optimum is normally body temperature, but it doesn't have to be. But the vast majority of enzymes have their optimum temperature at 37 degrees C. This graph here probably shows us that this enzyme might be an enzyme that has been engineered to work in a high temperature environment by humans. So this could be an enzyme that's used in washing powders, biological washing powders. The enzymes are sort of more stable and they work at higher temperatures because we've changed them that way. Or it could be an enzyme that's been isolated from some sort of organism that lives maybe in a hot spring. So that might be something called an extremophile. That's a bacteria that lives in a bubbling hot spring, and it has evolved to have enzymes that work at higher temperatures. Okay, so that's normally 37. This one is actually at 55. That is possible. So I can just write maybe here, but can be engineered or / I'll just write the word extremophile. Oops, oh, file. Okay, all right, so what's going on in this graph? How can we explain the shape of the graph? Well, there's two parts to this graph. First of all, this part. So on the upslope, okay, so increasing temp increases particle movement. As those particles are moving around more quickly, there is a greater chance that the substrate molecule is going to come in and fit into the active site. That's going to happen more frequently, so therefore, as we increase the particle movement, there's more collisions between enzyme and substrate, more enzyme-substrate complexes forming. That's the key thing that you've got to say. So I'll put that there. So increasing in ESC, okay? ESC stands for enzyme-substrate complex. More enzyme-substrate complexes forming, and therefore a faster reaction. But over here, this is happening, okay? Over here, the temperature has become too high for the protein structure. So as the protein, remember, is a string of amino acids linked with peptide bonds, but then coiled on itself. Now that coiling, the string itself is not going to come undone. The amino acids are not going to break. The peptide bonds generally won't break. But as the temperature increases, that coiling up starts to get disrupted. So it unravels. The protein becomes denatured. So here, this is the 3D protein shape. And here, the shape is unraveled. We don't use the word unraveled. We have a specific word for that. Of course, you know it. It is denatured. So what is happening when something is denatured? It's basically the bonds that hold the structure, the 3D structure of the enzyme, start to get broken. So what type of bonds have we got? On this diagram here, actually this internal red mass is showing hydrophobic interactions. Typically we have, well, always we have in globular proteins, hydrophobic interactions in the core of the protein that hold it together. And then we can have hydrogen bonds and hydrophilic interactions on the outside. But when the temperature is high enough and there's enough kinetic energy, so let's put that here, Ke, enough kinetic energy shaking the molecule about, then the whole thing's going to come unraveled. It's going to unfold and it may even clump together. So for example, when you cook an egg, what happens? Let's just do a brief application underneath, okay? What happens when you cook an egg? Well, when you cook an egg, I'll just do a little red, each globular protein in the egg white, okay, is separate, okay? The proteins in egg white are called albumin proteins, and they're floating about in solution separately like this. That means that the egg white is clear. So let's just kind of show that you can, I don't know, sort of light can go through it. You don't have to add this if you don't want to. But when you cook it, when you cook an egg, what happens is all those little albumin proteins, they unravel. And then they start to tangle together. So when you cook an egg, this little protein here kind of unravels, and they all unravel, and it becomes a massive tangled mess. So egg white, as we know, when we cook it, it's white. It was clear to start with, then it becomes white afterwards, and that's because in its unraveled state, basically light can't go through it, it becomes opaque. So this is clear, uncooked egg white or albumin, and this is white cooked egg white, and that's due to denaturing. Okay, so denaturing causes the enzyme not to work, and you've got to be specific. Let's not forget to say why. Denatured, the active site will no longer fit the substrate. So therefore it won't work. Now, it's fair to say that it's really in this region here, if you look where I'm pointing, this region is denatured. This region is increasing the temperature around this optimum, just right there where I'm pointing now. If we go to, let's just say, just a teeny bit above the optimum temperature, we're not really in the realms of denaturing yet. At that point, it's just that the active site is ever so slightly kind of, I don't know, maybe wobbling because of the temperature, and it's not quite a perfect match, okay? Once we go too far beyond, that's when it becomes denatured. We can describe this relationship also by something called the Q10 relationship. So over here, let's use that. So over here on the upslope, we can talk about Q10. So the Q10 coefficient means, well, it's a way of measuring the kind of gradient of the slope there. And so let's say we were looking at, if you look closely here, the temperature's 30 and 40 for this enzyme, okay? This enzyme, remember, is a higher temperature enzyme than normal. So between 30 and 40 here. So Q10 is equal to the rate of the enzyme at some temperature T plus 10 degrees, okay? Divided by the rate at the temperature T. So, for example, if we were working it out between 30 and 40 here, it would be the rate at 40 divided by the rate at 30. So that T in this case here would be 30. So 30 plus 10 would be 40, and then that's just the 30. So we're basically saying, if we look at any enzyme and say, okay, it's working at this speed at one temperature, if we were to increase it by 10 degrees C, what would it be working at that temperature? How much does it go up? And typically we find that in most enzymes, it's around 2 up until the optimum. So as we increase the temperature by 10 degrees C, the rate of the enzyme reaction doubles. Okay, so that is what the Q10 coefficient means. But if we were to go beyond the optimum, then obviously that relationship would not occur. So that relationship only occurs on the upslope. Okay, so that I think is everything to do with temperature. Okay, so now let's talk about another thing that affects enzyme activity, and that is, of course, pH. Okay, so pH, first of all, what is it? We need to understand what it is before we can talk about how it affects enzyme activity. So pH is really, if you want to get chemical, this is not really on the A-level biology syllabus, but chemists amongst you will know that pH is minus log 10 concentration of, well, H+, let's say. So it's a measure of the concentration of H+. So at low pHs, we've got high concentrations of H+ up here. And at high pHs, we've got low concentrations of H+. So it's all about the H+ ion and how it can affect protein structure. Here is an example of what secondary structure element that we've learned about in protein structure. Can you think? Hopefully you do. Remember, it's the alpha helix. So this could be an alpha helix. And this is an alpha helix at, let's say that's at pH 7. Okay, neutral pH. Now if we add H+, that's what it says right here. Let's make it orange. H+ added. Okay, so we're taking it to a lower pH. Now we can, well, it's not, oh, here we are. Yeah, now we have these H+ ions. There's one, there's two. And those H+ ions, if you look closely at this diagram, you can see what they're doing. Let's zoom in a little. What they're doing is they're interacting with charges that were holding this alpha helix together. So before, we can see that there's interactions between, let's just highlight these positive charges, I'll underline these in red, and the negative charges, which I'm going to put do with a little green underneath one. Okay, so these are basically hydrogen bonds between the negatives and positives within that chain. And what happens if we have lots and lots of hydrogen ions added is those hydrogen ions start to interact with the negatives on the chain, replacing the interactions that are holding the alpha helix together. That can cause the 3D structure of the protein to change, just like with temperature. Also, so that's one thing. So let's talk about that. Okay, so that's disrupt hydrogen bonds. Another thing it can do, pH, is it can alter the R groups. So sometimes proteins have an R group that is an ionically charged. So for example, they may have a carboxyl group, so C double bond O, and then another O over here. So hydrogen ions can change the ionic R groups. And alter the ionic interactions. Also, you can have hydrogen ions kind of what's called clustering around negatively charged ionic R groups. So that's another sort of version of what we can see here. This is kind of a version of clustering. So basically that's going to disrupt the structure. So what does that mean? Well, it means that if a given enzyme has an optimum of 7, okay, if we change the pH a little bit to the side, if we add a bit too many hydrogen ions and we take the pH lower, or if we remove hydrogen ions and we take the pH higher, that's going to affect the activity of the enzyme. So let's just label that up here. So let's say small changes. Oops, that's too big. Small changes to pH reduce the activity. I'll try ENS for enzyme. Okay, and that's because the active site is no longer perfectly the right shape. Kind of close, it's not perfectly the right shape. Now for small changes either side, if we then return the pH back to the optimum, there will be no long-term effect. So it's only a very temporary shifting away from the perfect shape. So you wouldn't really use the term denaturing, but it's a temporary change in the enzyme shape. For larger changes, it can permanently denature the enzyme. So large pH changes denature. So that's over here, also over here as well. So at D and E, we have denaturing. So I'll just write denature there as well, denature. Okay, now one thing it's worth remembering about pH changes is that enzymes and also proteins, because enzymes are proteins, they can have different pH optima is the plural, sorry, of optimum. So for example, if we zoom in a teeny bit, and look down here, here's two different enzymes. So you might get asked in an exam what are the different optimums. So if we put this kind of line down here, trypsin has an optimum of about, well, I don't know, I think it's about 7 to 7.5 from memory, I think. Pepsin, about 2. So why is it that enzymes can have such different pH optima? Well, it's really because they're designed, or they have evolved, to work where they need to work. So these are two digestive enzymes. Both of them are protease enzymes, but they work in different places. So trypsin in small intestine, which has a very slightly alkaline pH because of the action of bile, which then neutralizes the stomach acid and takes the pH just ever so slightly alkali, whereas pepsin is in the stomach. And in the stomach, we have hydrochloric acid. So in the stomach, it is about pH 2. So it has to work with the stomach acid. Now, in terms of the structure of the enzyme, you don't need to know so much detail, but basically the structure of the pepsin enzyme has evolved to be stable at low pHs. So it expects there to be a lot of H pluses floating about in order for its structure to be stable. And actually, if you take away those H plus ions floating around, then it would be denatured. So pepsin is actually denatured at pH 7. At neutral pHs, pepsin is fully denatured. You could see that here if we were to look at this curve going down, 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 down. It would be fully denatured at around pH 4 or 5, and at 7 it's definitely not going to work at all. So that is something to be aware of. Okay, so that's pH, another key factor that affects enzymes. Now let's move on and talk about some other enzyme graphs that you need to kind of analyze. Okay, so the three graphs that we need to look at show how the enzyme activity changes under certain conditions. And let's first of all label each of these kind of graphs that we need to look at. So first, this is a substrate concentration one. What happens to the enzyme as we increase the substrate? This one is to do with an inhibitor. And this is actually competitive inhibitors. Oops, inhibitor. And the bottom one here is going to be a non-competitive inhibitor. inhibitor. Can't write inhibitor today for some reason. Inhibitor. So now let's zoom in and let's look at the top one. So first one, if we increase the substrate. Now, I think at this point it's worth making a brief side note. Okay, whenever we're doing enzyme reactions and we're looking at how substrate affects the rate, we always look at the initial rate of reaction. Okay, because in an enzyme reaction, as we start the reaction, the enzyme breaks down substrate. As the enzyme breaks down the substrate concentration gets lower and lower and lower. So therefore, the enzyme reaction starts very quickly, and then the rate of reaction slows until it completely stops when the substrate is used up. So we normally do an initial rate of reaction type of method. So let's kind of actually just take a little bit of a side note and talk about that here. Initial rate. So we might, for example, have a test tube. This is just a little experimental method. We might have a bung in our test tube, and we might have, I don't know, potentially in here, we have some hydrogen peroxide, H2O2. Okay, so that's the substrate. And we can change the percentage concentration of H2O2. Okay, and in there, we might put some pieces of living tissue. It might be cubes of potato or pieces of liver even, and that contains the enzyme catalase. Then we could connect this up via a delivery tube, and we could connect it, let's say, to a gas syringe, okay? So the gas syringe would measure the rate, a badly drawn gas syringe there, it would measure the rate of oxygen produced. Okay, let's just label that because you probably can't tell what it is. Gas syringe. And that would measure oxygen produced in centimeters cubed. O2 cm cubed. You'd have to give it a fixed time interval, so there may be like per minute or maybe per 30 seconds. Let's say per minute. Now, obviously, if this was a scientific investigation, the thing that we'd be changing in this investigation is the changing the percentage of hydrogen peroxide. And of course, we need to keep constant certain things like the volume of this liquid, also the sizes and the pieces of the biological tissue, the type of tissue. Let's say we're going to use potato, same type of potato, same mass, and roughly the same surface area as well. And probably even better would not be to use pieces, but to do a puree of it, because it's more controllable. Okay, so what type of results would we get from this? We'd have to plot a graph. Over here would be the volume of O2 produced in cm cubed, perhaps. And over here would be time. Might be seconds, might be minutes. Now, what we would typically get is we would get an increase in the rate of oxygen being, we would get, not an increase in the rate, we would get the oxygen being produced quickly at the beginning, and it would slow and slow and slow and slow and slow and then stop. The reaction would then stop. That's supposed to be horizontal with respect to my axis, okay? Stop. And then to measure the initial rate, we would have to use a tangent. So you'd get your ruler lined up with this line. So the tangent, I'm going to try and draw it here. It's not going to be very good. Oh, it's done that all right. And then we could measure the rate here by taking the y change, this change over the x change, and working out the initial rate. Okay, so let's just write that. Use tangent to get initial rate. Then we would actually plot the initial rate, okay? Initial rate would be plotted over here. So we plot the initial rate of reaction on this graph. So this is reaction rate here would be an initial rate of reaction. All right, tangent finished. What does the actual result look like as you change the substrate concentration? Well, pinch in a little bit, we can see the result and we can also explain it. So here, the substrate concentration is the limiting factor. So as we increase the substrate concentration, the initial rate of enzyme reaction increases. It increases, it increases. But after a point up here, increasing substrate has no effect. Okay, so that is because all of the active sites are full. We should say occupied, actually. All of the active sites of the enzymes are occupied. So if there's a substrate molecule that's waiting to fit into an enzyme, it kind of has to wait its turn, wait for an enzyme to release its product, and then it can fit into the enzyme active site. So we can see here in this diagram, we've got all these substrate molecules sort of waiting, lined up for these just 3 enzyme molecules here that are ready. Whereas earlier on, when we were increasing the substrate from a low concentration, every time we added more substrates, it meant that there was more chance of a substrate entering the enzyme active site and forming an enzyme substrate complex. So over here, we would say substrate conc is the limiting factor. And if you increase the substrate, then what happens is you have an increased chance of collision and more enzyme substrate complexes forming. Whereas up here, you can't form any more enzyme substrate complexes because your enzyme is the limiting factor. Also, you get a very similar graph if you increase the enzyme concentration. Okay, so a similar graph would be very, very similar if we were increasing the enzyme over here, and this was the rate. I'll just label it R. The shape of the graph would be exactly the same for very similar regions. Okay. Now let's talk about inhibitors. My penmanship is not very good today. I don't know why I'm struggling. Now let's go down here and talk about inhibitors. Now you've got to recognize this graph. This is such a key exam question type thing. This graph here, right? If you see that, immediately in your mind, competitive inhibitor should flash. Okay, let's explain why this graph is like it is. So the green line is a rate of reaction without inhibitor. Okay, without inhib, that's supposed to say, and here it is with an inhibitor. With the inhibitor. Notice that on this x-axis down here, we're again looking at increasing substrate concentration. And again, this would be the initial rate of reaction. So what that means is when you add an inhibitor to the enzyme, and you do the same investigation we just did before, it basically brings the rate of reaction down. Why does that happen? Well, with a competitive inhibitor, that's here, it binds into the active site. So the competitive inhibitor is this little yellow thing. This is the substrate, as are these over here. So when we have the competitive inhibitor added, the competitive inhibitor and the substrate are competing for the same active site. That's why we call it competitive. They're both trying to get into that active site. Sometimes the substrate molecule will win, will pop in there, will be broken down. Sometimes the competitive inhibitor will win, will pop in there, will sit there for a little while, and then will come out again. So this effectively reduces the concentration of the active sites that are available for reactions. That's why it reduces the rate of reaction. But what we can do to get over this inhibition is if we increase the substrate, so this should be increase in substrate, many times it will outcompete the inhibitor. So this is like over here, low on, let's say over here, there's a big change, big difference between the without inhibitor and with the inhibitor, because let's say, you know, there's a 1-to-1 ratio. We've got sort of, let's sort of kind of label it over here. Let's sort of say at this point, we've got a kind of 1 substrate to 1 inhibitor. Can you see what I've done there? So I've written 1S to 1I. So we've got an even ratio, but up here, if we increase the substrate really a lot, we might have something like 100 substrate molecules to 1 inhibitor molecule. And there's a competition there that's still happening, but because we've got 100 times more substrate molecules than inhibitor molecules, it's much more likely, we can see down here, that the substrate pops into the enzyme and we have that maximum rate of reaction, which we would have had without an inhibitor, can still be reached. Okay, so that's the key thing. With a non-competitive inhibitor, if we scroll down, with a non-competitive inhibitor, the graph looks different. So this is with the inhibitor. And this is without. Again, this would be rate. And remember always that it's probably initial rate we're talking about. So non-competitive inhibitors, they don't compete for the active site, they bind somewhere else. So here we can see an enzyme. Here's the substrate, here's the enzyme substrate complex. Now, the non-competitive inhibitor binds over here. So this is at a different site. We call it an allosteric site. So that's not the active site. Allosteric is not the active site, it is somewhere else on the enzyme, but you can sort of see what happens here. If I sort of trace out this shape of the enzyme here, the enzyme here has this kind of rectangular up-down shape like that, okay? Whereas if we put this non-competitive inhibitor in the allosteric site, then the enzyme has a change of shape. Can we see that? So now the active site will not fit this molecule. So that is how it functions, okay? So binding of the I'm just going to write I for inhibitor, binding of the inhibitor changes the shape of the whole enzyme and specifically the active site, so that it won't bind anymore. Okay, the substrate will not bind anymore. And the key thing about non-competitive inhibitors is once they bind, they basically stay bound. Okay, so they bind pretty much permanently, and that essentially just takes out a proportion of the enzyme. So it's like reducing the number of enzymes that are there, by let's say 50%. So that's why the rate has dropped from up here to here. And adding more substrate won't do anything because the substrate is not competing directly with the inhibitor. Okay, so that is why this graph looks different. Right, okay, I think that's everything. So, quick recap. Enzymes, biological catalysts, proteins, very specific 3D shape. They can be anabolic, they can be catabolic. They can work inside cells, intracellular. They can work outside cells, extracellular. Wherever they work, they always speed up chemical reactions by lowering the activation energy. There's two different models. One says that the enzyme kind of has a really fixed shape, that's lock and key. One says the enzyme slightly changes shape as the substrate molecules bind, and that's called the induced fit. Enzymes can be affected by lots of things. Temperature and pH both affect their activity by essentially changing the shape of the enzyme, and that can either be with too much kinetic energy, kind of shaking the protein apart and denaturing it, or it can be with hydrogen ions affecting the interactions that hold the protein together and changing its shape again. Graphs that you need to be aware of: how does enzyme activity get affected by increasing the substrate concentration? It rises, it rises, it rises, then it plateaus as all the active sites are full. How does a competitive inhibitor lower the rate of enzyme reaction? Well, it lowers it a bit, but it can be removed by adding more substrate. Whereas with a non-competitive inhibitor, once you've added that inhibitor, we just reduce the rate, and you can't get rid of that inhibition by adding more substrate. Final little thing is sometimes we can use things like a non-competitive inhibitor for something called end product inhibition. Okay, so end product inhibition, last thing I've just thought of, sorry, this is a badly drawn diagram, I'm trying to rush. So basically here we have a sequence of enzymes acting. So this is the very first substrate molecule here. It's processed by one enzyme into this molecule, processed by another enzyme into this molecule, processed by another molecule by another enzyme into this molecule. So let's call these molecules A is the very first initial substrate, B, C, and then D is the final product. Sometimes the end product, D, might act as an inhibitor for this very first enzyme represented by the red arrow. And that basically is a way of the cell controlling the concentration of the final thing. That might be end product inhibition. Okay, so that is now everything. I hope that's been useful, and then I'll be setting, the next thing I'll be setting for you is some questions, multiple choice, on this enzyme stuff. Thanks very much, and I'll see you later. Bye.